Hi everyone, it's Mrs. Bustamante. In this video, we're going to talk about how do eukaryotic cells undergo regulation, right? How do they control what gets expressed or not expressed, right? Because we know most cells have the same DNA, but we also know that not all cells are exactly the same, right? Different genes can be turned off and turned on at different times, and that gives us differentiation in gene expression, or rather, different genes get expressed in different types of cells. Eukaryotes are going to have 5 main levels of control. 3 of them are going to be in the nucleus, and 2 of them are going to occur in the cytoplasm. You can see them listed here, and basically you'll notice it's how we go through the process of transcription and translation, or to make those proteins. Generally speaking, eukaryotes don't have operons, right? So we can't regulate them in the same fashion, but the genes can still be controlled together because a lot of times they share the same control elements between genes. So we can control one gene at the same time as another, just as we saw in operons. So why do we want to be able to turn genes off and turn genes on, right? Well, that's really just because of specialization, right? Different cells we know have different jobs, which means we have to use different genes in order to get the right job, if you will, out of that specific cell. And we call that specialization, right? They're kind of keyed in or tuned into a certain job, right? This can happen during development, right? Different genes oftentimes are needed to be turned on or off at different points in the cell cycle of an organism, right? Sometimes we need a certain hormone or a certain protein expressed to deal with something, and sometimes we don't. So we can turn it off afterwards, maybe just for a short time or even permanently at some time. The organism can respond to needs, right? The whole goal is to maintain homeostasis, right? This nice balance. Sometimes we need this, sometimes we don't need it, right? And that allows cells to basically be able to maintain that nice balance. And cells of multicellular organisms must continually turn certain genes on and off in response to signals from the external and the internal environment in order to do all of these things. So why is specialization good, right? Well, we have our body is big, for example, and we have lots of different types of cells, and we have lots of different things that need to be accomplished, right? So we want cells to have a specialized job because that kind of means that they can do that specific job better, right? So we want them to become differentiated or specialized. By the way, undifferentiated cells are what we call stem cells. There are some they just occur, right? They're cells that wouldn't necessarily have a programmed job yet. Sometimes we call these programmed cells, but stem cells we kind of think can become anything, if you will. So groups of cells that have the same function we call tissue, right? And this presence of specific proteins within the cells of tissues give the tissue its specific function, right? So I don't often look at my skin and think like, cool, skin cells, right? I'm talking about the tissues that make up my skin as I know it. Phenotype of a cell or organism is determined by the combination of genes that get expressed, right? So how those genes get turned on and turned off influences how they get expressed, and that's our phenotype. So our first level of control is our chromatin structure, right? We talked about DNA can sometimes be coiled up into chromosomes, but we also talk about that we need to have it undergo transcription, right? We need to be able to have RNA polymerase access it and make an mRNA copy. And how tightly wound that DNA is really controls whether or not this can happen. And we do that by kind of chemically changing these histone proteins that wind up our DNA, right? And the DNA, I guess, itself. So we can have what we call DNA methylation, which is the addition of methyl groups to the DNA, and this causes it to be more tightly packed, and it's inaccessible when it's in this form, and we call it heterochromatin. However, we can also have histone acetylation, which adds acetyl groups to the amino acids of the histone proteins, and that makes the chromatin less tightly packed, so that's going to kind of encourage transcription and make it more available. We call this euchromatin. We can see this in kind of another picture here, right? Here's our euchromatin and our acetylation on it, and you can see that these histones are sort of spread apart more, right? Versus methylation, you see the methyl groups on these histone proteins and they're close together or clumped together. We can also see this in real life. The darker DNA is chromatin, where it's more tightly packed, and the loosely packed one is euchromatic and it's lighter. So this leads to something we need to talk about briefly called epigenetics. Epi actually means above, if you will. So these are genetics or things that influence the expression of genes other than just the genes themselves. Really, epigenetics is the study of inheritance patterns that do not depend on the genes themselves, right? So kind of how can we make modifications, or how are modifications made that influence how genes are expressed, right? Inheritance patterns that are reversible based on modifications of the DNA or the histones, right? When it's tightly packed or not, how does that influence which genes get expressed, right? Could that change with the environment? And that's just the idea of epigenetics, that there's more than just genes themselves influencing phenotype. So our second area of control is in our transcriptional control, and this is probably the most important in eukaryotic regulation. Transcription factors are proteins that assist RNA polymerase in binding to the promoter. Really, their job is to help RNA polymerase find the promoter. In eukaryotes, we notice that there's what we call a TATA box, and it's a conserved sequence of the promoter. It looks like this. It looks like TATA. That's why we call it the TATA box. We find it in pretty much all eukaryotic organisms in the promoter region, which provides evidence, right, of evolution, of common ancestry, because we see it passed down, down, down. And really, it basically just helps the RNA polymerase find the specific region where it should attach and go, and because it's the same in a lot of places, right, we can make it easily recognizable. And you can see it down here too. There are also transcription activators. These are proteins that bind to the enhancer region, typically upstream of the promoter, and you'll notice that it kind of helps make like a little loop here, so that RNA polymerase can more easily access and synthesize mRNA. Then we have some post-transcriptional control. mRNA has been made, right? We're going to do our splicing of introns, our linking of exons. We're going to add our 5' cap and our poly A tail, and then we're going to make our mature mRNA, right? So how is this controlling it? Well, we're only using certain parts, right? Only activating certain parts. Our mRNA processing as part of our post-transcriptional control after that mRNA is made, and turning it into mature mRNA. So there are also things called these small RNA molecules. Some RNA molecules that exist in the cell can actually inhibit or help transcription and translation. Typically, they form double-stranded mRNA, like they kind of fold together, and it can regulate what mRNA sequences or what other sequences reach it to the ribosome to get transcribed. A lot of times they can interfere with it, which we kind of stumbled upon through some research, and destroy different mRNA sequences, things that look kind of funny to the cell, if you will, or don't look normal. It can kind of attack them, destroy the mRNA sequence, and so it won't reach the ribosome, so you won't see it expressed as a protein. This is often called interfering RNA. We see it as RNAi or microRNA or small interfering RNAs. All things that kind of disrupt the mRNA from reaching it as another way of regulation. We can also look at the lifespan of the mRNA, right? How long does it last? Can last anywhere from hours to weeks. Basically, how long does it need to stay around in order to continue to make that function? And then we can block the initial stage, right? Regulatory proteins attached to the 5' end of the mRNA and prevent the attachment of ribosomal subunits An initiator tRNA block the translation of the mRNA to protein, right? So something interferes with it, so it can't actually bind to the ribosome and be transcribed. Or excuse me, translated. Be translated. Then finally, we have our post-translational control. Okay, my protein is made here, but now what, right? Well, remember, we have to have functional proteins, and so we only have functional proteins that remain, right? Active. Did they fold correctly? Did they cleave? Did they add sugar groups? Are they targeted for transport, right? Basically, did they get made correctly so that they can function, right? Misfolded proteins we know would be problematic because structure determines function and it wouldn't work the way you wanted it. So proteins can also be degraded. We can do two things, ubiquitin tagging or proteasome degradation, right? The cell limits the lifetime of normal proteins by selectively degrading them or breaking them down. Many proteins, such as cyclins involved in the regulation of the cell cycle, must be relatively short-lived. Ubiquitin is a small protein present in all eukaryotic cells that participates in the destruction of defective proteins and synthesis for new proteins, right? So we don't want proteins to be around too long. We can tag them with these small proteins that would cause them to be degraded, and we can make new ones. So that's how eukaryotic cells control gene expression and cell specialization. I hope this was helpful, and have a great day.